Hi friends, welcome to the channel The Nurse. Here we are discussing about upcoming NIMHANS and JIPMER nursing exam practice questions. So here I am starting with the microbiology subjects. So later we will see one by one subject question practice questions. So first we will see microbiology questions. So we will move to the topic. Before that, if you are not subscribed to our channel, please subscribe and don't forget to click the bell button. So first we will see microbiology MCQs 2023 for preparing NIMHANS and JIPMER nursing officer exams. This is our first part. So also you can see our previous questions and answers for JIPMER and NIMHANS practice questions. So that is previous year's preparation videos. So you can see that also. It will be helpful for you, those who are preparing for these exams. So this is a fresh batch of questions. And first question, here I included some simple questions and some medium level difficult questions also. So some people may feel it is very easy for them. So those who are having this kind of problems, they can skip the video. Others can start the video, and it will be helpful for preparing more questions. So first question, which of the following is not a type of bacteria? Gram positive, gram negative, acid fast, eukaryotic. So here correct answer is eukaryotic. So what is eukaryotic? There is two types of cells, eukaryotic and prokaryotics. Eukaryotic means that is specified with the proper structures. The cell structure will be properly arranged. Those cells are known as eukaryotic. Eu means normal, normally arranged things, everything. So prokaryotic means it is not well arranged. So that kind of cell structure is seen in usually single cell organisms like bacteria, protozoa like organisms. So here I will read the descriptions. Eukaryotic cells are not bacteria. Bacteria are prokaryotic cells, which means that they do not have a nucleus or other membrane bonded organelles. So inside the cells there will be membrane bonded cell organelles. So that means the cell will be eukaryotic, the properly arranged cell structure. In bacteria, it will not be properly arranged. That is why it is known as prokaryotic cells. So examples of cell organelles includes nucleus, endoplasmic reticulum, Golgi apparatus, mitochondria, plastids, lysosomes, and vacuoles. These and all we can see specifically seen in plant cells or multi-celled organisms like humans or animals. So these are the peculiar features of multi-celled organisms, that is eukaryotic. Prokaryotic means that cell organelles is disorganized. Second question, which of the following is most common type of bacteria? Gram positive, gram negative bacteria, acid fast bacteria, eukaryotic bacteria. So in this question, you can easily exclude these options, eukaryotic. In previous questions, we have studied bacteria. Bacteria is an example of prokaryotic organism. So this D option you can exclude here. Only three options you can see. So correct answer is gram negative. Common type of bacteria, they are gram negative. Gram negative bacteria are most common type of bacteria. They are found in wide variety of environment, including the human body. So I will give you some examples for gram positive and gram negative bacteria. First, we will discuss. We will give examples for gram positive bacteria. So gram positive means the after the staining, it will retain the stain color, purple, violet color. They will retain. Then it is known as gram positive bacteria. Those bacteria not able to retain the staining color, so it is known as gram negative bacteria. So first we will see gram positive bacteria, Staphylococcus, Streptococcus, Listeria species, methylene resistant, that is MRSA bacteria, Streptococcus agalactiae. Then we will see gram negative bacteria. First is Enterobacter species, Salmonella species, typhoid causing bacteria, Pseudomonas species, that is mainly pneumonia and all causing bacteria, Shiga, E. coli, UTI causing bacteria, Chlamydia, Neisseria. These are the examples for gram negative bacteria. So next question, Which of the following tests is used to identify gram-positive and gram-negative bacteria? Acid-fast chain, gram stain, culture and sensitivity, polymerase chain reaction, or PCR? Correct answer, gram stain. It is an easy question. The gram stain is used to identify gram-positive and gram-negative bacteria. The test was by staining the cell wall of the bacteria. That means we will put a staining material over the slide. So, and after that, there will be a wash. Then after the wash, if the bacterial cell wall is retaining the color, it is positive. That is gram positive bacteria. If it is not retaining, it is called gram negative bacteria. Gram positive bacteria have a thick cell wall that retains crystalline violet stain, while gram negative bacteria have a thinner cell wall that does not retain the crystal violet stain. Fourth question, which of the following tests is used to identify acid-fast bacteria? Acid-fast stain, gram stain, culture and sensitivity, polymerase chain reactions. This also easy questions from the question itself you can answer here. So correct answer is acid-fast stain. The acid-fast chain is used to identify acid-fast bacteria. These bacteria have a thick waxy cell wall that is resistant to most stains. However, they can be stained with a special acid-fast stain. So these acid-fast bacteria are resistant to most of the all other stains except acid-fast stains. So it will retain only acid-fast stain color only. In that way, we can identify. The most common type of acid-fast bacteria are Mycobacterium tuberculosis and Mycobacterium leprae. One is TB causing, one is leprosy causing bacteria. So it is easy to remember these bacteria, acid-fast bacteria, Mycobacterium tuberculosis and Mycobacterium leprae. Fifth question, which of the following tests is used to identify the species of bacteria? Acid-fast stain, gram stain, culture and sensitivity, complete blood count. So here correct answer is culture and sensitivity test. During the culture test, we can isolate the which organism, which species of bacteria present in that specimen. Culture and sensitivity testing is used to identify the species of bacteria and to determine which antibiotics the bacteria are sensitive to. So after the culture test, we can identify the bacteria. Then after identifying, we can do the sensitivity test to identify which antibiotic is more sensitive to that particular organism. Then the test involves growing the bacteria in a laboratory and then testing the bacteria against different antibiotics. That means incubating the bacteria in the controlled environment in the laboratory setup, and it will take around 48 to 72 hours. After that, the particular bacteria will grow in that particular broth or culture media. So after identifying the colony structure and all, we can identify which bacteria are present in that specimen. Then we will do sensitivity test. Then we can identify which antibiotic is more powerful to destroy that bacteria. Then which of the following test is used to identify the genetic material of bacteria? Acid-fast stain, gram stain, culture and sensitivity, polymerase chain reaction. To identify the genetic material, we can do polymerase chain reactions. Polymerase chain reaction in COVID time and all, we used to do that RT-PCR. So RT means real time, PCR means polymerase chain reaction. Polymerase chain reaction or PCR is used to identify the genetic material of bacteria. The test works by amplifying a specific sequence of DNA. PCR can be used to identify bacteria even if they are not alive. So this will be helpful to find bacteria also, not only virus. It will helpful to find bacteria also. In previous question, we have studied that culture test to identify the species of bacteria. So in culture test, it is only useful if the bacteria is live. If bacteria is already died because of the external atmospheric difference or other unfavorable environment, the bacteria may die. So even after the death of the bacteria, we can identify with the PCR test, polymerase chain reaction. Then seventh question, which of the following bacteria is the most common cause of urinary tract infections? E. coli, Staphylococcus aureus, Streptococcus pneumoniae, Neisseria gonorrhea. Correct answer is E. coli. It is an easy question. It is a very repeated question. So you should remember this answer. So same questions will ask in most of the all government exams. Common cause of urinary tract, that is E. coli. Escherichia coli is the most common cause of urinary tract infections. The full form of E. coli, Escherichia coli. Eighth question, which of the following bacteria is the most common cause of pneumonia? Streptococcus pneumoniae, Haemophilus influenzae, Mycoplasma pneumoniae, Legionella pneumophila. Pneumophila. So most of the all organisms will cause pneumonia, but most common cause of pneumonia, that is Streptococcus pneumoniae. So in previous question we have studied most common cause of urinary tract infection. Here we have studied most common cause of pneumonia, that is Streptococcus pneumoniae. Streptococcus pneumoniae is the most common cause of pneumonia. Which of the following bacteria is the most common cause of food poisoning? Another thing is food poisoning. So here option is Salmonella, Campylobacter, Listeria, monocytogenes, E. coli. Salmonella is the most common cause of food poisoning. Usually that is cause food poisoning in adults. But most common cause of food poisoning in children, that is Clostridium species, especially Clostridium botulinum. In adult, most common cause of food poisoning, that is Salmonella. Salmonella, it is an interesting organism. Salmonella is the most common cause of food poisoning, and mainly Salmonella has two species, Salmonella enterica and Salmonella bongori. Same time, the Salmonella enterica is the type of species and is further divided into 6 subspecies. And that 6 subspecies include over 2600 serotypes. In that serotypes, Salmonella typhi, that is typhoid fever causing organism, that is one serotype. So Salmonella enterica is divided into 6 subspecies, and this in that subspecies there are 2600 serotypes, different serotypes. So one of the serotype is Salmonella typhi that causes typhoid fever. There is another paratyphi also there. So it will cause paratyphoid fever. 10th question, which of the following bacteria is the most common cause of sexually transmitted infections? Neisseria gonorrhea, Chlamydia trachomatis, Mycoplasma genitalium, Treponema pallidum. Most common cause of sexually transmitted disease, that is Neisseria gonorrhea, which causes gonorrhea. Neisseria gonorrhea is the most common cause of sexually transmitted infection. It causes gonorrhea. 11th question, which of the following is not a type of sample that can be collected for medical testing? Blood, urine, stool, brain tissue. Correct answer is brain tissue. But to collect brain tissue, we need to do surgery, or that is correct. To brain tissue is not a type of sample that can be collected for medical testing without a surgery. And all other samples we can collect without a surgery, but brain tissue we need to collect after a surgery only. 12th question, which of the following is the most common type of blood sample collected? Venous blood, arterial blood, capillary blood, fetal blood. It is another easy question. So correct answer is venous blood. Venous blood is the most common type of blood sample collected. It is collected from a vein in the arm or leg. 13th question, which of the following is the most important step in urine sample collection? Cleaning the genitalia, collecting a midstream sample, refrigerating a sample immediately. All of the above. So here correct answer is all of the above. All steps are important for collecting urine sample. First we need to clean the genitalia. Then we need to take the midstream urine only. We need to instruct the client to collect midstream urine. Avoid first voiding and last voided urine. during the midstream, patient has to or client has to collect urine sample to get a accurate report and reduce the contamination. And immediately after collecting, we need to refrigerate the sample if the delay is there to transfer the sample to laboratory. Otherwise, if it is keeping outside means the organism present inside the specimen will die. All of the above steps are important in urine sample collection. Cleaning the genitalia helps to prevent contamination of the sample. Collecting a midstream sample helps to ensure that the sample is representative of the entire bladder. Refrigerating a sample immediately helps to preserve the sample. If there is a delay in transferring, we need to refrigerate the sample. 14th question. Which of the following is the most important step in stool sample collection? Collecting a sample from multiple sites, avoiding contamination with the urine, storing the sample in a clean container, all of the above. Correct answer is all of the above. While collecting a stool sample, it should be collected from multiple sites, avoiding the contamination with the urine, storing the sample in a clean container. All of the above steps are important in stool sample collection. Collecting a sample from multiple sites helps to ensure that the sample is representative of the entire stool. Avoiding contamination with the urine helps to prevent contamination of the sample. Storing the sample in a clean container helps to preserve the sample. 15th question. Which of the following is the most important step in saliva sample collection? Rinsing the mouth with water, collecting a sample from sublingual gland, storing the sample in a clean container. All of the above. In this question also, all of the above is the correct answer. All the above steps are correct only. All of the above steps are important in saliva sample collection. Rinsing the mouth with water helps to remove any food or debris that could contaminate the sample. So important thing is the rinsing with plain water only. No need to use any antibiotic solutions. Collecting a sample from a sublingual gland helps to ensure that the sample is representative of the entire saliva. Storing the sample in a clean container helps to preserve the sample. 16th question. Which of the following is not a type of sample processing technique? Centrifugation, filtration, extraction, amplification. So correct answer is amplifications. So amplification means it is a type of nucleic acid testing technique. It is not a processing technique. Amplification is the process that is a type of nucleic acid testing technique, not a sample processing technique. In question, we have asked the sample processing technique, not a testing technique. Then 17th question, centrifugation is a sample processing technique that is used to separate the different components of a sample, concentrate a sample, remove impurities from a sample. All of the above. So it is one of the sample processing technique. So correct answer is all of the above. So in centrifugation, the uses of centrifugation that separate different components of a sample, it will concentrate a sample, remove impurities from a sample. Centrifugation is a sample processing technique that is used to separate different components of a sample, concentrate a sample and remove impurities from a sample. Then 18th question, filtration. Filtration is another sample processing technique. It is a sample processing technique that is used to remove large particles from a sample, sterilize a sample, concentrate a sample, all of the above. Correct answer is all of the above. But in this filtration technique is not that much useful for sterilizing a sample, but main purpose is remove large particles from a sample and it helps to concentrate a sample. The larger part will be differentiated from smaller particles. So that will help to concentrate the larger particle in different area and smaller particle in different area. But the sterilization method that is not useful that much, but still it is used in earlier days, but nowadays it is not used. Filtration is a sample processing technique that is used to remove large particles from a sample. For an example, if large impurities are there means we can differentiate the impurities and we can filter out the smaller ones. So smaller ones may be bacteria or other things. So we can easily find out the smaller bacteria from the smaller particles. It can also be used to sterilize a sample, but it is not a very effective way to concentrate a sample. Concentrate, I told to it helps to concentrate the sample, but it is not a very effective way to concentrate the sample. So best way is centrifugation. Extraction is a sample processing technique that is used to. It is another third one. Extraction. It is used to separate a specific substance from a sample, concentrate a sample, remove impurities from a sample. All of the above. Here also correct answer is all of the above. So I just kept these questions for understanding purpose only. Extraction, centrifugation, filtration, these are sample processing technique. You need to aware that. So that purpose only I kept these questions here. So extraction also, in extraction means it is particularly specific. It is separating specific things in a different area. So for an example, it will separate the microorganism, extract the microorganism or extract the particular things from a mixture of solution, and it will helps to concentrate a sample. So it will remove the impurities from a sample. So it will from the mixed sample, it will remove the impurities. Then the sample will be perfectly included with that microorganism or that researcher's interest. Extraction is a sample processing technique that is used to separate a specific substance from a sample. It can also be used to concentrate a sample and remove impurities from a sample. Then today's last question. Amplification is a nucleic acid testing technique that is used to detect the presence of specific nucleic acid sequence in a sample, quantify the amount of specific nucleic acid sequence in a sample, characterize a specific nucleic acid sequence in a sample, all of the above. So in previous questions, we have learned that amplification is a testing method, testing technique. It is not a processing technique. So amplification means it will amplify, it will make the things in a larger size. Amplify means it makes a larger size. So like a lens, we are seeing small things in a larger size. Amplification process also, it's like that kind of mechanism it is doing. The smaller nucleic acid, it will make a larger size. Then it is easy to find out which organism having that particular structure of nucleic acid. So in that way, we can identify the organisms. So one of the common test that is used, this amplification test is CBNet. So you can tell the or you can write the full form of CBNet test in our comment sections. So I need to see who would are my correct answer. CBNet usually used to identify mycobacterial tuberculosis, pulmonary tuberculosis in children. Nowadays it is used in adult also. So kindly please comment what is the full form of CBNet. So here correct answer is all of the above. Detect CB amplification is helps to detect the presence of specific nucleic acid sequence in sample. So by assessing that specific kind of nucleic acid sequence, we can identify the organisms. It will quantify the amount of specific nucleic acid sequence in a sample. Quantify means it will make same copies of the DNA sequence, and it will multiply that copies. So the persons can easily identify, or the machine can easily identify the nucleic acid sequence. Nucleic acid amplification test, commonly known as NAT, is a molecular diagnostic technique to detect the genetic material, that is nucleic acid, in each specimen sample. So the PCR test is one of the example of amplification test. So there are different amplification tests. In that, CBNet is one of the tests, and RT-PCR or PCR, polymerase chain reaction, is another type of amplification test. So that is about today's questions. So if you have any doubt, means you can ask in comment sections. So I will come with another video regarding NIMHANS and JIPMAR practice questions. So Till then, you can watch our previous videos also. I have made a separate playlist for NIMHANS previous questions. So you can watch it there. So thank you and please stay tuned. Bye bye.